So we were discussing like new chapter which was development and aging. Two last videos were done like fertilization and intro of that chapter. Today we are going to see cleavage and egg types. So let's see. Here comes the cleavage, and you'll also see egg types we'll discuss. So first thing, let's discuss cleavage. Cleavage is simply dividing of the cells. Like, what does this mean, dividing of the cells? Division of the cells. So before when I start, cleavage and mitosis somewhere considered the same, but we have a major difference which I would love to tell you now. So let's see here. When we talk about the cleavage, this is simply when during the mitosis of embryo this happens. This means cell division in the embryo. Cell division in embryo happen. That's called as cleavage. Then this can be only seen in animal cell only, and in this one cytokinesis process happen, like something cell division, and that's all. When we talk about the mitosis simply which occurs cell division of parent cell into daughter cell. Here is the parent cell, then division to daughter cell, called as mitosis, parent cell, daughter cell. And you'll see this happen both like plants and animals both. And the last one, cytokinesis plus karyokinesis, both process occur in this one. So I'm sure it will be clear, and this is the difference. And if we like for today, we'll discuss cleavage. So division of cell simply, or cell division in the embryo as well, occur only in the animal cell, or cytokinesis process occurs simply, which I've explained. If I simply tell you the major difference, like what is cleavage for you, not for you, but of course is a genuine cleavage is what? It's simply if this the zygote, as we know, a zygote is formed by the egg and sperm fertilization, so that's called a zygote or fertilized egg. After that, what will happen after this what will happen, you'll see other divisions like this have become this called as blastocyst. After this, not blastocyst but blastomere. After this blastomere, this is going to form other 4 structures. Right after the structure, you will see more than like 16 structure which are quite loosely packed. Here we call 4 cells. Here this called as morula. So cleavage is what? Cleavage is simply the division when the cell are dividing. Here we know zygote was 1, but it formed 2 more structures. Then from this one we formed 4 more structure here. Then 8 cell. Then again 16. So, but we see the division again and again, and even these all division. If we just talk about blastomere, this was zygote, right? When it get divided, so the gene information is present not only in one part but even in the whole part. Like all here are 2, but in this 16 genetic information will be provided in all, right? So here we'll see simply when zygote get division to other smaller cells, zygote divide to other smaller parts, and even they all have their genetic material called as zygote. And zygote again have a few things like our topic is about cleavage and eggs, so we'll discuss both today. So I'm sure cleavage is clear to you. Simply smaller cell, even when we'll read more you'll get it more nicely, so I'm sure it will be clear. So let's see. Okay, I think picture have become quite bigger, so let's see directly from the PDF. So here comes the PDF. Let's read what's written. So cleavage and egg types. We'll discuss cleavage is what? Process of rapid cell division occur after fertilization. So as I mentioned, here's the egg and egg and sperm, when they both get fertilized, there will be rapid cell division. And what will happen during with zygote, which divided into smaller parts, smaller cells, and each contain a copy of the genetic material from the original cell. As I mentioned, when zygote is formed by what? By fertilization. After fertilization, the zygote got divided to in smaller cells, and then each contain a copy of genetic material from like original cell. So I am sure it will be clear. The purpose of cleavage is to generate many cells that will eventually form the tissue, organs, developing embryo. So the main purpose is simply because when cleavage would be there, this means we can generate so many cells, so many not organs but so many cells, and which will even have the same genetic information. So then they will gradually help. We know when cell combines, they form tissue, so they will gradually increase and form the tissues. Then from tissue it will form organ, then from organ it will form organ system, like that. So developing of embryo will helps. The amount of yolk present in the egg cell can significantly impact the cleavage process. If this the egg, we know, right? This egg, the amount of yolk is the yellow part. The amount of yolk which is present in the egg cell will significantly impact. This means if we have a small amount, it will impact. Even if we have large amount, it will even impact. So like that, I'm sure it would be clear. Then in animal species such as birds, reptile, egg contain a large amount of yolk. This means they're telling birds, reptile, they have a large amount of yolk, which we can make a cleavage more difficult. So then cleavage become more difficult in those which have the large amount of yolk. I'm sure it will be clear. Then let's see. In eggs with moderate to little yolk, cleavage occurs throughout the whole egg, pattern called as holoblastic cleavage. Like in eggs, we know if there's a moderate or a little yolk, so cleavage occurs throughout the whole egg. When it occurring through the whole egg, that term called as holoblastic cleavage. However, an egg with a large amount of yolk, as like we know birds, reptile have a large amount, with a small amount of clear cytoplasm and concentrated one pole called as blastodisc is simply I'll show you picture. But before that, if if this okay if this the diagram on the here, you'll see this is called as blastodisc, and rest other which only have a yolk like structure, this form concentrated structure here, this called as blastocyst. I'm sure it will be clear. Then cleavage in these eggs restricted to the blastodisc. Here these eggs restricted to blastodisc. The yolk is essentially an inert mass, and when this yolk is inert mass, the type of cleavage pattern is called as hemeroplastic. So this type of cleavage where yolk is essentially an inert mass called as meroplastic cleavage. So I'm sure it will be clear. Let's see the diagram, how it's dividing and everything. Or okay, let's discuss hopefully. So here are holoblastic, or we even call this as a isolated radial. This means this is present in the radial echinoderms and amplurus. Here's the one cell zygote divided into two blastomere, then four, then this forms 16 structure called as morula. In the second one, we have spiral and leaves mollusca flatworms. In this one, they are telling about kingdom Animalia, which animal come in this one division. So here Annelida, mollusca, flatworms, earthworms is all. The structure you can see here, the more cells. Then we have bilateral tunicate, one, then blastomere, then four, then more than not four, these are 16, then so many. Then rotational mammals. We have so many other structure. Then melochial. Then we have another layer called meroplastic, which is simply bilateral like a cephedo mollusca. Here we get division inside. Then we have dissolved fish, reptiles, bird, like this structure. The last centralithial, like you can see the structure. So I'm sure it will be clear. We have these X type, which we'll discuss in another video hopefully. Thank you for watching. Bye bye, stay connected.